In this webinar, we'll be taking a look at BioCyc tools for navigating metabolic and regulatory networks and for analyzing omics datasets. I'm Peter Midford, a member of the Bioinformatics Research Group at SRI International. This series builds on the material presented in the Introduction to BioCyc webinar series, and I recommend you start there first if you are not yet familiar with BioCyc. In part 1 of this series, we'll be looking at the cellular overview, the metabolic map diagram for a given organism. Although this diagram is mentioned several times in the introductory webinars, here we will be looking at it in more detail. We'll start at the BioCyc homepage, and before opening the overview, we'll verify in the upper right of the web pane that E. coli K12 EcoCyc is the selected database. Once we have the correct database, select the cellular overview by choosing it off the metabolism drop-down menu. Note that a unique cellular overview diagram has been generated for each organism database in BioCyc to contain its pathways, reactions, and transporters. And that takes us to the cellular overview diagram for E. coli. Several tools let you control this diagram. On the left is the zoom level control and indicator, which should be familiar from the Genome Browser webinar in the introductory series. On the right is the operations menu, which is similar to operations menus for other displays, and an overview legend, which hangs off the left side of the operations menu. We won't need these to get started, so I will hide both of them. There are also 3 slider controls at the top of the display, which control the look of the diagram in ways I will briefly discuss later. Now let's take a look at the diagram itself. Let's start with how the diagram is organized. Most of the diagram consists of small node and line networks that represent sets of reactions. Nodes represent compounds, while lines, also called edges, represent reactions. If you look closely, you'll notice that nodes come in different shapes and some are filled. These shapes communicate the chemical classes of compounds that a node might represent. There's a legend that explains these shapes available in the operations menu. Let's open the menu and take a quick look at the legend. The minimized menu is on the upper right, and I can click to open it and find Show Legend near the bottom. The legend window lists kinds of compounds and their corresponding icons. It also tells us that filled symbols indicate the compound is phosphorylated. The legend can be dismissed via the button or the close box at the right of the title bar. I'll minimize the operations menu as well. The diagram is likely too large to fit in your browser window, so you'll need to pan it. To do this, just hold down the mouse somewhere where there isn't a reaction, then hold the mouse button and drag. If a window pops up, it should close by itself momentarily. Likewise, you can zoom the diagram in and out with a mouse wheel or the equivalent gesture on your trackpad. Note that the zoom indicator on the left side of the window will show the level you have zoomed to. You can use its buttons to zoom in smaller discrete steps than the mouse wheel provides. Let's consider the overall layout of the diagram. Metabolic pathways are collected into functional sets, such as carbohydrates biosynthesis and detoxification. Anabolic pathways are collected on the left. Catabolic pathways are on the right. Energy-producing pathways are in between. You can see that fermentation and respiration are here. There are also a large number of individual unclassified reactions to the right of the catabolic pathways. There's also a pair of brown lines running around the edge of the diagram. These represent the plasma membrane and the cell wall respectively. The paraplasmic space is between the two, and reactions that occur in that space are gathered at the far right of the diagram. There are also transport reactions that move metabolites across membranes. So let's take a look at a compound within a well-known pathway, the TCA cycle. The cycle is depicted as a circle over here, and at about 5 o'clock on the circle we find 2-oxoglutarate. If I mouse on it, and wait, a window pops up that identifies the compound and provides several ways to find out more about it. If I click on the name or the ID of the compound, it takes me to the compound information page in a new tab. Likewise, if I click on the name of the pathway, it takes me to the pathway information page. There are 2 buttons on the Windows title bar, one to close the window and another labeled Keep. This button keeps the window open if you move the mouse away. Holding the window open also replaces the Keep button with 2 buttons labeled T and P. Each of these buttons will, if clicked, shrink the window by filtering the contents. The T button shows only the title or name of the compound. The P button shows only the pathway. The shrunken window retains the T and P buttons. Clicking the same button in the shrunken window a second time will revert the window to its full size and contents, while clicking the other will switch to its filtered content. Now let's zoom in a bit. If I slowly zoom in, I eventually get to the point where I only see a portion of the TCA cycle, but as the drawing gets bigger, there comes a point where new details appear. The first detail to appear is the name of compounds appearing in the backbone of the pathway. If I continue to scroll inwards, the names of the pathways themselves appear in dark boxes. Continuing to scroll, the last labels to appear are the enzymes and their associated genes for each visible reaction. This is as far as it goes. If you look over at the zoom control on the left, you'll see that the slider is nearly at the top. While we are here, let's have a look at the enzyme that catalyzes the production of 2-oxoglutarate in this pathway, isocitrate dehydrogenase. Again, I just mouse over the name and we see a similar pop-up with links for the name and pathway, as well as the name of the gene that codes for this protein without a link. Before zooming out, let's take a look at the reaction catalyzed by isocitrate dehydrogenase. Moving the mouse over the reaction line, a pop-up similar to the one for compounds appears. The reaction window includes the enzyme and information about its reversibility. If we use the Keep button to hold it open, we see 3 buttons to filter the contents. T and P operate as they do in the compound window, and the E button just shows the enzyme. Also, if we move the window away from the reaction, it will actually draws a line linking the window back to the reaction. Now let's explore the right sidebar operations menu. I'll zoom out first. There are 3 sections to the operations menu. The first is the Experimental Data Overlay, or Omics viewer, which will be covered in the next part of this webinar series. The bottom section covers utility commands. We've seen the legend already. The help command takes you to the section of the online documentation relevant to this overview page. Finally, generate bookmark generates a URL string that you can paste into a browser to return to this display, including any highlighting you may have displayed. You can see the location and zoom level are encoded in the URL. That leaves the highlighting operations for finding items of interest in the diagram. Highlighting paints over a compound or reaction in a highlighting color. It should make it stand out in the display. You can highlight pathways, reactions, genes, enzymes, or compounds. For each kind of thing that you can highlight, there are multiple ways you can specify the particular individual or subset of the kind that you want to highlight. All kinds support searching by full name or entering a substring, which will pop up a list of choices. The more characters you provide in the substring, the shorter the list. Let's try a few simple examples that will show what happened when we highlight a pathway and then a set of reactions related to the product of that pathway. I'll start by finding the pathway for L-tyrosine biosynthesis. To do this, I will click on the Highlight Pathways portion of the menu, and once it's open, select by substring. Type in window appears, and I will start typing TY. And now I see a menu of options, and I can choose L-tyrosine biosynthesis 1 and click Highlight. Now over on the left, the pathway for L-tyrosine biosynthesis is painted over. Each reaction is shown in green. If I open up the Overview Layers legend, it now lists both the underlying cellular overview, but now also the pathway name, rendered in the same color as the highlighting. With a highlighted pathway, I can demonstrate the use of the slider controls at the top of the window. With the highlighted pathway in view, if I slide the opacity control to the left, everything in the diagram becomes more transparent except the highlighting. When I get all the way to the left, everything in the diagram except the highlighting becomes transparent. Moving that slider back, if I move the middle slider, the reaction lines or edges become thicker or thinner. I'll leave them a bit thinner, and now I can thicken the highlighted edge lines with the third slider. And that makes the highlighted edges thicker. So these controls are a way to tailor visualization to best display the highlighting on the diagram. Let's add a little more. It's reasonable to expect that somewhere in the tyrosine synthesis pathway, there are likely to be one or more reactions inhibited by the presence of free tyrosine. Let's see if this holds. I will now switch and I will now open the Highlight Reactions submenu, and here we see, in addition to the common ways of specifying a set of reactions, that we can choose by modulator. If we open that, we see choices of cofactor, activator, or inhibitor. Let's open the inhibitor submenu and choose by substring again. I type TYR. There will be more of these because there are more compounds than reactions or pathways, and find L-tyrosine on this list. I choose, click the highlight button. Now I see red overlaid over several reactions, not just the synthesis pathway we previously tagged, but let's zoom into the synthesis pathway. to the compound level to see just where free tyrosine is inhibiting the pathway. And what we see is that there are multiple reactions in the pathway that are regulated by free tyrosine. The first reaction is in the charismate pathway, as well as several steps that are closer to the final tyrosine product. Note that tryptophan synthesis is not inhibited downstream of charismate synthesis. You can also see a tyrosine inhibited step in the neighboring pathway to the right of the tyrosine synthesis path. Of course, we can zoom in a little closer if we want to browse the details of the enzymes. Now, what can we do with the overlay legend window? First, we can turn the highlighting in each set on and off. Another thing I can do is to display a list of all the reactions in the highlighted set. Because I'm logged in, I can create a smart table from the list of the reactions, which might be useful if we wanted to dig into this further. I'm actually not as interested in this pathway as the set of reactions inhibited by tyrosine, so I'll dismiss this list and bring up the list for the other set of free tyrosine inhibited reactions. And just for now, I'll save it as a smart table for later. Well, that concludes this highlights tour of the cellular overview in BioCyc. We covered the layout of the cellular overview, how to navigate it and drill down for more detail, and how using highlights lets you ask interesting questions about regulation of pathways. I hope you have found this useful. Thank you for listening.